Hi, everyone. Welcome to the second training sessions of the Changer project. Thank you very much for being here. I am Alice, and for these sessions, I'm actually more the moderator. So I will accompany through the session. I will introduce the various presenter, and I will moderate the case discussion, but I will not present today. So this session will be on gene editing, but before starting with the topic of today specifically, I think it's useful to point out that these training sessions are part of a bigger project, the Changer project, which is a EU-funded project under the Horizon Videra scheme. And the whole scope of Changer is to support ethics review in innovative technology. And this specific training is part of a series of training. We already had one previously on electronic informed consent. This one is on gene editing, and we will have two coming in autumn on AI and organoids. So just a practical information, this session will be recorded and the recording will be published on our own Changer project page. So for today, sorry. So for today, I think someone has the mic on. If please you can turn it off. Thank you. Okay. So the agenda for today, we will start with a general introduction on gene editing. And then we will dive a little bit deeper on the legal landscape, and we will focus more specifically on the European setting. And then we will have a little break. I think it will be needed. And we will come back with the core of our training that is going to be the ethical landscape and the ethical discussion surrounding this technology. And we will conclude with a case study, which will require some engagement on your side. The idea is that we will present a case, and then we will divide in breakout rooms and have a really smaller case discussion to try and better analyze the case, also based on your experience, but especially on the presentations that we saw today. So now I will stop sharing. That works out. And I will introduce the next speaker. Okay. Hey, sorry again, I can't stop sharing. Can you? Okay, do that for me. Yeah, perfect. Pascal's not sharing. Okay, it's perfect. Thank you. It's solved out. So the first speaker is Professor Pascal Borry. He is a professor of bioethics at KU Leuven, and his focus is, broadly speaking, on the ethical, legal, and social implications of new technology. He is also a member of multiple ethics committees, so he has a lot of both theoretical and really hands-on practical experience, and is also the chair of the WADA Independent Ethics Board. Thank you very much, Pascal. Floor is yours.
Okay, thank you very much for this introduction, and it is indeed my pleasure to somewhat introduce the topic and the field of gene editing, and maybe the introduction seems somewhat focused here on the more technical part, but I will say something about the technical part, but of course I want already to engage more on the ethical and societal implications of a new technology that got, I would say, more traction in the last 10 to 15 years. So I think, having worked a lot in the domain of human genetics, I think what is a striking, I would say, tension that has existed, that a lot of investments have been done, first of all, in trying to understand the human genome. Think about the International Human Genome Project. I mean, a lot of investments was really like, can we understand the basis of the conditions, pathologies? But if I think more broadly, if I think about organisms and plants, cells, animals, it's just about what is the function some of the codes that are in our genome. And what has always been the idea, of course, it's one thing to understand the next step. What you would like to do is, in order to be able to correct, to adapt, if you know that a certain mechanisms in your genome, whether it's a deletion, whether it's a repetition, whether it's a kind of a mistake, a letter that is mistake, a snip that is mistake, that you can adapt and correct that. That's ultimately what we would like to do, and or what researchers would like to achieve for. And of course, the question is, if you start to doing it, what kind of concerns does that bring up? And a lot of discussion about gene editing has been developed from the moment that the new technology came into the attentions around 2015-16, CRISPR-Cas9, with the idea like, oh, well, we can adapt in a cell by kind of even a word processing. It's like a find and replace function. I think it's a nice way of writing it. Is that you are kind of repairing a defective DNA strand with a healthy copy, and that's the goal idea of gene editing. What of course I would like to emphasize first of all is that when we talk about gene editing here, it is really about a precise, permanent DNA change that we are doing in a cell or in an organism. And there, I think if we go a bit further in the rest of the module and the discussions around gene editing and moreover gene therapies, you would see that this is a bit more complex, because it is not always about permanent DNA changes. And I think, for example, about a lot of the gene therapies that we have are not always working with a permanent DNA change. You have as well RNA-based therapies that are working more at the RNA level, where changes are not permanent, where you work with gene additions, where you make a change separate from the genome. Think about Zolgensma, which is a gene therapy for SMA, which is a little bit different than what I present here. So just don't want to go too much into the technical part, but I think that's important as well when we come to cases and when we come to reflections about to what extent the intervention is at the genomic level, or is it more at the protein level, for example, RNA level, and that has an impact as well. But as a starter, I will focus here about how gene editing is clearly defined. It's a change at the genetic level, which has a lot of advantages. Is that you can kind of correct a mutation, you can insert a gene at a specific locus of the genome, you can delete, disrupt problematic sequences, and you can apply that to any kind of cell type. Focus in the rest of this seminar will be more on the human applications. I will tell a bit more about plant and other applications as well on organisms. But the idea here is this is a permanent effect, and tools are relatively simple and really accessible to all kinds of scientists. And I think that's really an interesting development, and researchers are using that just for example to in animals to mimic diseases by integrating genes, so that whether they know whether therapies would work in specific organisms. So that's how technologies are being used. Of course, if we talk about gene editing and more broadly, before I go to more specific applications, well, we know, of course, that this is something that is happening in nature spontaneously as well. Think about developments in organisms. You have changes that adapt, and over generations, you see sometimes changes happening as well. And of course, it brings ethical issues. And just to give you an example, that has been debated a lot over the last 20, 30 years. Genetically modified organisms, to what extent this is artificial compared to nature interventions, and think about applications for example, about, can we use that to make plants have more fruits, can grow better, having variations in colors, and other examples that are mentioned there are all kinds of applications of, in fact, a similar technology that is being used. I want to start as well to just set the scene about something which is a little bit very ethics related, but where I would say we'll often not come even or reach even to an ethics committee. And that's a case we have been working on about to what extent you can use a gene editing tool, for example, to combat malaria. When we look at malaria, we know that this is a condition caused by a parasite, Plasmodium. And I'm not an expert in malaria, just to that you know, but I just want to focus on the application. Where you see that, in fact, it's that parasite that is being transmitted by mosquitoes, by very specific mosquitoes that are the vectors of that condition. And we know in order to combat malaria, different strategies are being used. And one of the most effective is that, of course, not being bitten by a mosquito. And you know that nets are being used to prevent attacks from mosquitoes. Insecticides are being used, reducing breeding sites for mosquitoes. And we know that a good socio-economic development as well is the best kind of way of fighting as well malaria. But what is interesting is that, despite as well all these efforts, malaria is still there. Investments were done for vaccines to target the parasite, was not very efficient. And then you see that the new projects were developed where the idea was like, well, maybe we could target the mosquitoes that are the vectors of that parasite. So, and that brings in a lot of kind of challenges as well. How do we deal with that? So we know that, for example, there are more than 400 species of that mosquitoes that are known. And around 30 to 40 are important as malaria vectors. And so we see that some projects have advanced the idea like, well, maybe we can genetically modify through a gene drive technology so that the pool of mosquitoes, in fact, is being changed. And where mosquitoes, in fact, what they are doing in some of these technical projects is making the female mosquitoes infertile. So that in lab environments, for example, population of these female mosquitoes of that specific species died after 7 to 11 generations, which is in a lab context. And of course, you see immediately the question like, oh, well, that's one thing to do that in a lab. It's another question about can you do that in a real life context, and what is the impact on that? And of course, at the ethical level, it brings quite a lot of questions. And I just want to raise some of them that were asked with regard to an experiment like that or an innovation like that. Because if you talk about a technology that leads to, in fact, an I would say an ending of a specific species is a question: what kind of impact does this create on biodiversity? Is that acceptable? Is that not acceptable? Mosquitoes will typically not be in life protection programs like we have in other for like lions or elephants. That's not really considered to be that we need to have a bigger protection, but brings quite questions about like, well, do we really know possible consequences of doing this? Is that a good strategy to do that. Some have argued that this should never be the first option, but rather a very last option if you would try to intervene in that way. What is the impact on the ecosystem? Because you know what you try to fight, but do you know the different consequences? Mosquitoes can as well be food for other organisms, so for animals. So what is the impact on your ecosystem if you are doing this? So it brings up tensions between, on the one hand, the precautionary principles that would argue you need to defend the, if you're not completely sure about the consequences, about the risks, then it's better to have a precaution position. Others arguing that this might hinder innovations because proving that it is completely safe is almost impossible. So, and others as well bringing up the tension about, maybe it's better not to innovate, to emphasize on in biotechnological innovation like this, but more working on the socioeconomic conditions so that you have other ways of addressing the problem of malaria. And then I didn't even talk about how do you engage communities that would be affected in this, and can you even have a controlled environment of this. So I think it's a nice case because it is really the question: how would you address ethical aspects of a project like this? Our project is focused on ethics review. If you think about ethics commissions, looking at research protocols, very much these are much smaller in scale. I think this is a nice case of a project with wider societal impacts, and where really you need the integrity, attention of the researchers, the engagement of societies, reflections and engagement with regulators, and so on. So that's one, I would say, field. I mean, example of an application. I would like to go a bit further now to cases we come closer to in our workshop here. Another strategy where gene editing could be used or has been used is a case in 2018 that got a lot of attention worldwide, where a Chinese scholar announced that he had genetically modified twin babies before they were born to make them resistant to HIV, which is a case of germline intervention. And just that you see here, it's the figure on the right, where you make a DNA change very early in development, and then which has an impact on all genes in your body, having an impact on the future generations. And that's very different than situations we will come a bit later on, which is a somatic cell gene editing, which means that you adapt specific genes in specific cells, but that change is adapted or limited, for example, to specific organs, and will not have an impact on future generations. So the idea of germline gene editing, when this case and scenario of that researchers came out, created a lot of worldwide discussions, concluding that the experiment was irresponsible, violated international norms, failed to meet ethical standards, did not have sufficient medical justification, lacked transparency, and these were just only some of the critiques that were given on that specific case, and it has to do with a lot of the problem that we didn't really know the impact that this intervention might have, the fact that the use of that intervention for this purpose was not really considered to be very useful as well, and creating a lot of risks as well. But what I think was interesting as well is that the system, to a certain extent, failed as well, is in that sense that there was not really a proper ethical evaluation that had happened that was able to to as well stop that case to happen. Anyway, it led to a moratorium, several documents that were issued raising that germline interventions, it's way too early these days. There are too many risks, too many safety considerations. There is not a legitimate purpose of doing this. And we acknowledge that not every country might have a legal framework that is sufficiently robust, but at least there are moral, ethical guidance documents that really called for a clear moratorium on doing this kind of interventions. So why, I would say, where then do we see gene editing approaches getting ground, I would say, and getting more approaches in practice as well, more close to an ethics review procedures in different ethics committees, for example. That's when we talk about the somatic gene editing and would call it more even, I think it's more correct than to talk about gene therapies, where you would have different approaches, as explained at the start, some that work more at the permanent DNA level, others that are working more at, for example, as well, non-permanent level, like RNA-based interventions. And there you can do that in vivo or ex vivo, where you modify the cells outside the body or rather inside the body. And what we see, this is CRISPR-Cas9 has brought an innovation, but this is not that it hasn't been there for, it has been there for a long time, gene therapies. And the technical problems that were always faced is that, well, you need to kind of use a vector to place the edit into your organisms. And then your immune system might react, provide resistance. You might have off-target effects, bringing a lot of questions about the potential benefits that you can do with a therapy, but the potential harms that can be created. So it brings immediately to ethics committees a lot of tensions about the harms that you can create by doing a clinical study in this context, bringing issues about who to integrate in a study, the selection discussion, the informed consent process about that, the potential therapeutic misconception in integrating people in such a trial. And we'll come back to all this topic later on. Maybe one example of a success story that is often being referred to, this is a case of a gene therapy that is linked to a very rare condition, often to rare conditions, here an eye condition that relates to blindness. And what is happening here is that a correct gene is being integrated into the patient's eyes through a vector, and this was happening quite successfully. But of course, this very often is a very expensive therapy as well, bringing a lot of issues as well about accessibility of treatment, justice issues. Why to invest in this, not in something different? And we come back to that. And of course, it's often for a small group of patients. Why do I want to bring this up as well? Because, we see that things are not always going well. There are clinical studies where we have seen that there was off-target effects where some patient died, and where there is a negative consequences for participants in studies, bringing again at the foreground that kind of really the difficult challenge for ethics committees about evaluation of such studies, because there are various studies taking place in different domains. With this, I wanted to just give an introduction of the topics, showing I would say, the spread of potential applications, some of the ethical discussions that arise as well, depending on the different applications that you can do. And I hope hereby opening a little bit the floor for next presentations. Yeah, thank you.
Thank you very much, Pascal. I think we have time for a couple of quick questions if someone has a question. Okay, if not, we can move to the next presenter. Thank you again, Pascal, for this
My
very
pleasure.
insightful and very necessary first technical background. So our next presenter is Margot Reckelbus. She has a background in law and population science, and she's currently a PhD candidate at KU Leuven. And her focus is, broadly speaking, on the ethical, legal, and social challenges of gene editing. But she has a particular interest on access, safety, and the regulation oversight, and especially on participants' perspective on these important topics. So today she's going to provide us with, again, another necessary background piece of the puzzle, that is the legal and regulatory consideration for patient access to gene therapy. Thank you very much, Margot, and the floor is yours.
Thank you, Alice. So hello, everyone. So I'm going to talk about legal and regulatory consideration for patient access to gene therapies, because when we talk about gene editing and gene therapy, it's important to understand that patient access is shaped by a complex legal and regulatory framework. So, unlike conventional medicine, gene therapies work by modifying, replacing, or introducing genetic material to treat or prevent disease. So because of their innovative nature, they raise unique scientific, regulatory, and ethical challenges. In the European Union, gene editing therapies fall under the broader category of advanced therapy medicinal products, or ATMPs. So this category includes gene therapies, somatic cell therapies, and tissue-engineered products. And because these products often involve genetic material, living cells, or highly specialized manufacturing processes, they are regulated differently from conventional medicine and require additional oversight. To bring an ATMP to market, developers must go through a centralized authorization procedure that is managed by the European Medicine Agency, or EMA. So rather than having each member state conduct its own scientific assessment, the evaluation is carried out once at the European level. So regulator assess whether the therapy is safe, effective, and manufactured according to strict quality standards. And within the European Medicine Agency, there is a specialized committee that is called the Committee for Advanced Therapy, or CAT, and it plays a very important role. So the CAT provides expertise on the scientific and technical challenges associated with advanced therapies, and it contributes to the overall assessment process. One big advantage of this centralized procedure is that the scientific evaluation only needs to be performed once for the entire European Union. So as a result, when a therapy receives marketing authorization, it can, in principle, be marketed across all European Union member states. So the aim of this is to simplify market entry, avoid duplication of regulatory work, and ensure consistent standards of quality, safety, and efficacy across Europe. So the system is mainly based on two key regulations, the ATMP regulation and the regulation establishing the European Medicine Agency centralized authorization procedures. So the ATMP regulation acts as lex specialis, so a specialized law. This means it overrides general European Union pharmaceutical legislation regarding advanced therapies unless otherwise specified. And for the regulation establishing the European Medicine Agency centralized authorization procedures, it implements the centralized procedure that requires pharmaceutical companies to submit a single marketing authorization application to the European Medicine Agency. And together, those two regulations form the foundation of the European Union framework governing gene therapy. So the European Union has also introduced several mechanisms to accelerate access to innovative therapies, so particularly for patients with serious or life-threatening conditions who have limited treatment options. So one example is the accelerated assessment. It shortens the review period for therapies considered to be of major public interest. There is another one that is called the conditional marketing authorization. So this allows a therapy to be approved on the basis of less complete clinical data than would normally be required, provided that the expected benefits outweigh the remaining uncertainties and that additional evidence will be collected after approval. And the EU has also created the PRIME scheme. So PRIME stands for Priority Medicines, and PRIME offers early scientific and regulatory support to developers of promising therapies that address unmet medical needs. So overall, these mechanisms aim to balance two important objectives: providing patients with earlier access to innovative treatment, while maintaining high standards of safety, efficacy, and quality. However, regulatory approval and accelerated pathways do not automatically translate into patient access. So despite these efforts, a significant barrier remain, and many patients still face difficulty obtaining these therapies in practice. So the first barrier is cost. So gene therapies are among the most expensive treatments that are currently available, with price often ranging from 1 to 2 million euro per patient. So even for higher income countries, integrating these therapies into routine healthcare budget can be extremely challenging. And for lower income member states, the challenge is obviously even bigger. So this raises important questions about fairness and distributive justice across the European Union. A second and third major issues are geographical inequalities and fragmented healthcare system. So although the authorization takes place at the European Union level, healthcare system, pricing decision and reimbursement policies remain national responsibilities. And as a result, some countries are much better equipped financially and institutionally to provide these therapies than others. So for example, patients in countries such as Germany or France often have easier access than patients in countries such as Romania or Bulgaria, where funding and infrastructure may be more limited. Importantly, these inequalities do not only exist between countries, they also exist within countries themselves. So for example, patients living in rural areas may face additional challenges because specialized treatment centers are often concentrated in large cities. And so this means that access therefore depends not only where someone lives in Europe, but sometimes even on where they live within their own countries. So taken together, high costs, geographical disparities, and fragmented healthcare system create significant ethical and practical barriers to access. Beyond the traditional authorization pathway, the European Union has also developed two alternative routes that can provide earlier access to gene therapy. So there is the hospital exemption and the compassionate use program. So hospital exemption allows hospitals to prepare and provide certain gene therapies on a case-by-case basis without the full European Medicine Agency authorization, provided that they comply with national requirements. So this pathway is often used in urgent clinical situations or for very rare disease where no authorized treatment exists. However, because implementation is largely left to national authorities, access also varies considerably across the European Union. So hospitals in higher income member states are generally more likely to have the expertise, infrastructure, and financial resources needed to use hospital exemption in practice. And as a result, important concern about equity arise. Then there is the compassionate use program. It provides another route to access. So this allows patients with serious or life-threatening condition to receive therapies that are still under investigation, particularly when they cannot participate in clinical trials. So again, with compassionate use program, access depends heavily on the willingness and capacity of national healthcare systems to implement and finance this program. So consequently, patients in lower income countries often have fewer opportunities to benefit from it. So although both hospital exemption and compassionate use are designed to expand access, their uneven implementation can sometimes reinforce existing inequalities. So this brings us to some of the broader limitations of the current system. So first, implementation remains fragmented across member states. So some countries have the infrastructure, expertise, and funding needed to support alternative access pathways, while others still struggle to do so. Second, affordability remains a major challenge. So even when therapies are legally available, their costs can make them inaccessible in practice for many healthcare systems. And third, centralized market authorization does not automatically guarantee patient access. So although approval takes place at the European Union level, each member state still negotiates its own prices and reimbursement conditions. And as a result, a therapy may be readily available in one country while remaining unavailable in another. So in other words, regulatory approval and actual patient access are not necessarily the same things. So many member states have therefore developed national strategies to manage these challenges. So although their approach vary considerably. So for example, in Germany, they have experimented with outcome-based payment models, so where the reimbursement is then linked to the clinical success of the therapy. So the idea is to reduce financial risk by ensuring that payment reflects treatment outcomes. The Netherlands has explored amortization and subscription-based payment models, so which spreads cost over time rather than requiring a single large upfront payment. But by contrast, other countries in the European Union often face greater difficulties because of more limited healthcare budget. So access may depend on individual reimbursement decision rather than structured national programs, and this restricts the number of patients who can benefit from the treatment. So even when therapies are formally reimbursed, additional barriers can remain, including underfunded hospital, shortage of specialized professionals, and out-of-pocket costs for patients. As a result, the effectiveness of national strategies often depends on both the economic resources and the organizational capacity of the healthcare system. And the challenges become even more pronounced in the context of rare disease. So to encourage innovation, the European Union introduced the Orphan Drug Regulation, which provides several incentives for companies developing treatments for rare disease. So these incentives include first, market exclusivities. So this means that for a certain period, competitors generally cannot market a similar product for the same indication. Then there is the reduced regulatory fees. So this lowers the cost associated with scientific advice and regulatory procedure. And then there is access to funding opportunities that can support research and development activities. So these incentives are important because developing treatment for very small patient population is often not commercially attractive. But at the same time, however, these incentives can contribute to monopolistic market condition. So when only one treatment exists for a rare disease, company may have substantial pricing power, which can lead to very high prices and place considerable pressure on healthcare budget. So while the regulatory framework is designed to stimulate innovation, it also raises important ethical questions about how to balance incentives for innovation with fairness and equitable access for patients. So to conclude, one of the main messages from this presentation is that authorization does not automatically mean access. So although the European Union regulatory framework has been successful in supporting innovation, many patients still face barriers related to affordability, geography, and national healthcare capacity. So moving forward, the key challenge will be finding a way to balance innovation with equitable access so that the benefit of gene therapy can be shared more fairly across Europe. This highlights the importance of recognizing that the ethical implication of gene therapies extend beyond clinical trial and into question of access, affordability, and fairness. Thank you.
Thank you very much, Petra. Sorry, thank you, Margot. Are there any questions?
Yes, hi. May I intervene?
Yes, yes.
Oh, hello, I'm Cynthia from Portugal. I just wanted to thank Margot because she has done a wonderful, brilliant presentation. So you have made a matter that is legally as difficult as you have seen, captivating, engaging, and clearly perceptible. So I would thank you for that. And I would ask you, because affordability, as you well said, is such a huge question, whether we could benefit in the future from a European strategy, not only for approval, but also for access, like negotiating capacity at European level in order to be able to compete and provide with equity and fairness to all people across Europe the possibility to give innovative treatments. Thank you.
Thank you very much. So for the moment, it's not in the agenda of the European Union, but I really hope in the future they can maybe, for very rare disease at least, place reimbursement at the European level, for example, so that patients with a very rare disease have access to treatment, even if it's not reimbursed nationally. But for the moment, this is not in their agenda. But they have some idea to improve access with, for example, the joint clinical assessment, or the fact that they want to harmonize the hospital exemption definition. So that's already something.
Yeah. Thank you so much.
Vicky?
Thank you. It's more a comment, actually complementing Cynthia's remark than a question. What Cynthia proposed is something that we've seen for the vaccines against COVID-19. So we do have a previous experience at the European level to kind of negotiate for possible vaccines. So why not for gene therapies as well? So this is a good idea that I think that at the policy level, it should be highlighted and examined. Another way to improve access to gene therapies that sometimes cost even millions of euros is that something that we've seen lately. So at the national level, governments actually make agreements with the pharmaceutical companies that develop the gene therapies, and they can reimburse them in installments. And there is even another criterion that is used. They have to pay only if the gene therapy has been successful. So I'm just adding this to the table because we've seen different formats of reimbursement that are coming out lately in the late years, just to increase the access to such gene therapies. Thank you.
Thank you, Rieke.
Any other questions? No, okay. If there are no more questions, then maybe we can start our break a little bit earlier. So we have 15 minutes of break. See you soon.
Thank you.
Thank you.
Thank you. We can start again. Yes, the recording restarted. So that's a sign that we can start again. Thank you very much for being here again, back from the break. So now we are passing to the next topic, that is the ethical landscape. And our first presenter is Rieke van der Graaf. She's also professor of bioethics, but at UMC Utrecht. She's also the head of the Department of Bioethics and Health Humanities. And her research focus is on global justice in international setting, fair selection of research participants, and the ethics of innovative research design. But broadly speaking, her main interest is research with human subjects, and she focuses on many different aspects, including inclusion of vulnerable participants, inclusion of pregnant women, and so on and so forth. And she will now focus more on what are the ethical aspects and the challenges when it comes to gene therapy. Rieke, thank you very much for being here. The floor is yours.
Thank you so much, Alice and Pascal, for having me today in your important changer projects. I like you to talk, not to all of you, you just introduced me for Alice, but to talk you to some of the ethics issues in research with gene therapy. I think there are ethics issues related to how to appropriately balance the need for speed and also the presumed delay if we do not approve these studies soon enough. To talk you through some of the issues related to curative gene therapies, issues related to placebo-controlled trials, fair access a bit, and informed consent. So it's a brief introduction to all of these topics which deserve more attention per topic as well. So the first topic of balancing speed and delay, I think what we can see currently is that many people are really excited about what science can achieve, not only scientists themselves, but also journalists and scientific journals. So, for instance, for hemophilia B, there is now Hemgenix, and that is said to cure people by major breakthroughs, and it's still having good results. The Lancet has also coined or dedicated its front page some time ago to all the advances in gene therapy for congenital blindness, for instance, and deafness. These all hold transformative potential. And last year there was big news around baby KJ, who was cured from his disease already at a very young age. Now, apart from all these, or just because of all these scientific advances, there's also a big societal expectation. And then some are saying, well, actually, Europe is falling behind because, there have been so-called ATMP approvals, so advanced therapeutic medical product approvals, primarily cell and gene therapies approximately by the EMA 20 approvals, and FDA has approximately approved 40 of these therapies, but you see that China and India are now new countries. And for instance, the Draghi report about the future of European competitiveness said, well, we have to accelerate in the innovation, we have to simplify regulation, and we have to do more R&D. Sorry, someone is in the waiting room. That's something else. Okay. There's also a report of FPIA about factors affecting the location of biopharmaceutical investments. And you see what the implications for Europe are. And you see that in Asia Pacific region, in the North American region, more investments are done around ATMPs and clinical trials. So what can we do in Europe? Like, should we simplify the regulation as suggested by the Draghi report? And I think to some extent that maybe in some ways that may be a good idea, but there may also be ethical issues. For instance, well, we could start from the assumption that health is a basic good. And that's something that you will see in these reports. And because health is a basic good, you also find this notion in various among various philosophers who have said, well, without health, there's no proper functioning, and it's difficult to pursue a life that's worth living. So if that's the starting point, then we could also say, well, if these products are being developed, then the products and treatments must also become available for Europeans, if that's your starting point. Another issue why we should follow this idea that we should simplify the regulation in Europe is that if we do not do it, then perhaps pharma and researchers may go elsewhere, and then they may relocate their research to countries with different levels of protection. On the other hand, you could say, well, there are these scientific breakthroughs, as I just mentioned, and there's also a way to manage the societal expectations, because we cannot get access to everything that is scientifically possible. So therefore, we also have to touch a little bit more on the debate of hope versus hype, which I will do later in the talk, and fair access. Now, there have been several procedural proposals as well, also for acceleration and simplify regulation, which I think are relatively feasible. For instance, harmonizing law and procedures. So there in Europe, we have the clinical trials regulation, for instance, and there have been various proposals to also simplify what's in the clinical trials regulation and in CITES. So the new Biotech Act has several proposals to try to speed up what we currently have. So for instance, the idea that we currently review clinical trials with requests for information, we have now 106 days, and this should be brought back to 75 days for review, and without protocols where are no requests for information, it should be reviewed within 47 days. And there are currently exceptions for rules for ATMPs, so we have 50 days longer. And that rule, according to the Biotech Act, if that's taken over, then it may be done minus these 50 extra days. So that's one way to speed it up procedurally. There are also alternative ways of product approval once the product is after the stage of a clinical trial and seeking market approval. So you could say, well, there may be conditional market approval, but then obviously you will still need post-marketing surveillance. That's a procedure that's allowed within EMA under strict conditions. And other alternative ways of product approval could be platform designation. That's something that is allowed by the FDA. So you have sort of one approval, and if there has been one approval, that may also be counted as approval for other techniques that are more or less similar, also under very strict conditions. But at the same time, there are ethical challenges if you're looking for alternative market approval. And I want to emphasize that, research ethics is important throughout the pipeline. And it starts actually at the moment of the design and goes on until the product is available. And it's not only something that we can and should do at the moment of research ethics approval of your protocol by your research ethics committee. And so therefore, I think as ethicists or within the ethics community, I think we should see a continuous challenge. Where is the line between fast and too fast? And something where I think ethicists should be taken on board in very early stages and also in the final stages of product development. And we should also look at the soundness of the review. That always takes time, and we should I think be aware that ethics is never a tick box exercise. So even if we can do it faster, we always need to do it carefully. And so I just want to remind you very briefly of where research can go wrong. And many researchers are really aware of when it can go wrong, and also believe that when you make mistakes in this process, it will have huge impact on further progress. And we have seen that in the case of Jesse Gelsinger in 1999. So there was ethics approval for this case. So this Jesse was included in a trial where he was actually the 18th patient in the study. And he was the second patient who was given the highest dose. He had a mild form of OTC deficiency. So he could be treated actually for that disease. It's a metabolic disease. And it's a serious disease, but he had a mild form, and there was gene therapy available for his disease. Now, there was ethics approval, including by the FDA and the NIH, sponsored the trials, and he gave informed consent, but he had just turned 18. But it turned out there were many mistakes, and eventually, so after administration of the injection with the gene therapy, he died. And then it turned out that there was also a huge conflict of interest of the co-investigator, and that the FDA was not informed about monkey studies that had been done earlier, or the grade 3 tox that was observed in other patients. And so he was not the first, but nonetheless, he died, and they could have known more about the previous patients who had experienced grade 3 tox. And I think, so nowadays, ATMPs are well regulated, but it's important to, I think, still see it as research. So even though there is, in European Union, we have centralized marketing authorization through EMA. There is the CAD committee, so the Committee for Advanced Therapies by EMA. We have the CTR, GMP, GCP. So there's a lot of regulation, and yet it remains research. And I think we have to be aware that things can go wrong, and that we have to take a careful look. And so my take-home message would be just not only at the moment of rec approval, but just very early on, and also until the product is available on the markets. Now, a little bit more about the curative gene therapies, because of the high promises. So cure is often used as a metaphor. So for hemophilia B, as I just mentioned already, but also for other therapies. And so with a PhD student in one of my previous projects, I looked at how we can define cure actually. And we found at least 2 different ways in which it is currently used. So one way of using the definition is normalization of the body, based on Christopher Bors's definition that disease is a deviation from statistically normal biological functioning, and health is then the absence of such a deviation. And the second is to see it as obtaining a normal life. So here Leonard Nordenveld has said, you are healthy if you are able to reach your vital goals. Now, we see those two definitions also reflected in the literature on hemophilia. And so Van Balen and colleagues have said, well, when can you say that someone is normalized? Well, normalized then means the clotting factors normalized clotting factor levels 5 years after curative treatment, and then cure is phenotypically intended. So that's something that you can then measure in terms of clotting factors. But if you look at that definition also for hemophilia B, you could wonder, so what should actually be normalized? Is it only those clotting factors? Should also the symptoms be alleviated? Or should we also cure any remaining symptoms? Should we prevent patients from getting further symptoms? So the treatment Hemgenix, for instance, which was so promising and miraculous, has now shown that there's durable disease correction throughout the 24 months after gene therapy for patients with severe or moderately severe hemophilia B. So that's very promising and stable. At the same time, we should also be honest and say, well, the safety and efficacy in the long term are still unknown. So there's still the risk of liver inflammation due to the immune response, and there may be limitations still for individuals with neutralizing antibodies. So yes, to a certain extent, I think we can say this is a curative treatment, but not for everyone, and there are still also uncertainties. Now, obtaining a normal life is a second way in which gene therapy is said to cure people. And for the context of hemophilia, where we did the research, we found a definition of a hemophilia-free mind. So meaning that the patient has no psychological burden or thoughts of hemophilia and complications, and is able to live a life without being confronted with a disease. And also the caregivers do not have to contemplate about the disease and the consequences of it. And that's sort of the goal that people wanted to achieve. Alleviating future suffering from the disease and the burden of its treatment, that's sort of the goal, and the hope of forgetting about the disease. But the doctors also that we spoke to also said, well, there's a risk that patients become reckless in a way, because they don't consider the need to be, if they are going on holiday, for instance, to be close to a hospital anymore. And that sometimes this being completely free is not always realistic. That's at least what the physicians that we talked to said. Now a little bit more about placebo. I think that's a third issue that is debated in this context specifically. If you look at the numbers, so for placebo and ATMPs, you see that many are still placebo-controlled, about 30%, or at least placebo or active controlled. Some are non-controlled and some are historical controls. Now, EMA and FDA also prefer randomized controlled comparative trials over single-arm trials or trials with external historical controls. And they say, well, where reference therapies are not available, comparison to best supportive care treatment based on investigator's choice or placebo is expected to provide evidence of efficacy and is preferred over single-arm trials. This was stated in their 2025 guideline. But if you look at the literature, you see many assumptions where people say, well, patients prefer studies without placebo when there's no standard of care, or patients prefer studies when the intervention is looking promising, and people feel it's unethical. That's something that we hear a lot, to provide patients with placebo in these trials if they do not have alternatives. So more methodologically, there are small sample sizes and an RCT is not always feasible. But I think ethically we have to pay close attention to what makes it unethical. And it's not necessarily the case. So if you look at a concept like clinical equipoise, that is a concept that is widely debated, but also I think among ethicists it's clear now that clinical equipoise is not a clinical equipoise in the mind of one single investigator and not in the mind of the patients, but something that should exist in the medical expert community based on the definition that of Benjamin Friedman, 1987, where he said, well, clinical equipoise is honest disagreement about the preferred treatment in the expert medical community. So it can be either two treatments that have been established where there's honest disagreement which of the two treatments is better, or there can be agnosticism, whether it's better or not than nothing at all. So where you have a situation where you can test to placebo. And so clinical equipoise is consisting of two separate underlying ethical requirements. So the one is this honest disagreement or agnosticism, and the second is that we should test whether patients are withheld from standard of care, meaning what is the established effective standard of care. So in hemophilia, it's not easy to test against placebo because there is an established practice of giving people clotting factors. And so it's not necessarily unethical to give participants placebo, and it's not inconsistent with Helsinki and SIOMS, but it's presumed to be unethical. And you can have a situation where participants prefer something else than placebo, but that doesn't mean that it's also unethical to provide placebo, especially not if you can fulfill these two criteria and if you can do a study that can accordingly lead to socially valuable knowledge. So I think the question that we do have to ask is what can be done to minimize placebo use if people do not want to participate in a placebo-controlled trial. So I think what we could do is look at the duration, the burden and the risks of placebo and sham, because obviously if you have to use a sham procedure, it can also be risky, and that's something that you can debate also in your research ethics committee. And sometimes it's just very burdensome to be in a placebo arm for a couple of years and undergo all these extra procedures. So that's something that you can address. And you can also use other types of designs, more modern designs like adaptive designs or designs with Bayesian statistics that can drop inferior arms sooner than if you use a regular clinical trial. But I think basically we should never expose people to research with low scientific validity and poor social value. I think if that's the starting point, because we want to gain more more knowledge, then we should not expose people to research with low scientific validity and leave the placebo out, because participants do prefer something else. A bit too on fair access. So of course, if you do your trial, then we know that gene therapy is costly once it is on the market. And it's not always reimbursed. For instance, Amgenix that I talked about. And there's also differences in access between countries in Europe. And I think Margot will talk about that as well. So the availability of the therapies is mostly via participation in trials. And that may create additional barriers so that you can only access it via trials because there are limited slots, or you may have to wait for results in adults. Specifically, if the trials with pediatric patients, then oftentimes it takes a while before the trial is started in pediatric patients, if at all. And there are also specific centers, for instance, where you may have to go to. So even though it is accessible via trials, not all people can access it or can participate in a clinical trial, and you have to meet the eligibility criteria in general, for instance. Now, it is so accessibility in Europe is a challenge. And I think a further challenge is that how we justify the access, because currently many of these trials are started from the presumption that there is an unmet medical need. or that there is a so-called rule of rescue or non-abandonment principle. But once there is a new product on the market, for instance, we have to see whether that still holds. So what actually should justify starting a new clinical trial, and is unmet medical need, is that the appropriate justification for it? Because if we have something to rescue people, that's probably no longer a sufficient justification. And here again, I think it's a continuum from starting a clinical trial to doing a clinical trial, or approving and doing a clinical trial, and then having the product on the market. And sometimes what we've seen, especially also with hemophilia, is that once the product is on the market, the participant may prefer something else, and then a very costly therapy has to be withdrawn from the market because patients and physicians are no longer interested, and that's also a waste of resources. So how do we actually justify the design, the conduct, and the accessibility in general? And I think what we have to pay more attention to also from a global perspective is the translational gap for gene therapies between high-income countries and low-middle-income countries. So most of these gene therapies are just developed with high-income countries in mind. And how do we actually get these gene therapies also developed from a more global perspective? And that's a debate on its own. But I think we need to pay more attention to that as well. And then the last topic I wanted to say a bit on is informed consent. So there's a big debate, obviously, on hope or hype, and many people also, what we found in our interview studies, hope have hopes, high hopes for hemophilia treatment, but hope is not always achievable. There's also a misunderstanding oftentimes that the gene therapy is just one sort of treatment, where actually there are, per disease and per condition, we need different therapies and different forms of the therapies, and some forms of these therapies are still preclinical. Then sometimes trials do not make it to the next stage, and not all patients are aware of what the latest results are. So what's realistic, whether it's still preclinical or whether it's already in phase 1 and the results are promising. So they've heard of gene therapy, but don't know when these results will reach the next stage, and also not whether this will actually someday will applicable to them or to another group. And so the question is actually, I think, how to educate people, and how can we have a very nuanced debate that tries to balance this hope or hype. And so for the Netherlands, we have made a documentary which we call Amending Heart. We also have English translation, and it will be available by the end of the year. So to try to have both the hope and hype perspective, and get that balance to have a societal debate about that. And the last thing I wanted to say a little bit on is the informed consent, and the so-called therapeutic misconception. So Paul Applebaum, the ethicist or psychologist, but also ethicist, has said, well, there are actually two forms of therapeutic misconception. And the one is an incorrect belief that a patient or the participant has individualized needs, that his or her individualized needs will determine the assignment to treatment conditions or lead to modifications of the treatment regimen. And then, or the second is the unreasonable appraisal of the nature or likelihood of medical benefit from participation in the study due to misperception of the nature of the research enterprise. And I think we see both forms of therapeutic misconception. And in a study already performed in 2005 by Jonathan Kimmelman and colleagues, he said, well, the consent documents used in the phase 1 gene transfer trials, although increasingly attentive to possible therapeutic misconceptions, are inappropriate, optimistic about the direct benefits of trial participation. So I think that's something in the hope and hype debate that we should pay attention to. So take home again, I think most important is to have well-designed and conducted research is the cornerstone of ethics, and that creates maximal social value and minimizes the risks of unnecessary exposing people to research risks. And I think we should involve ethics and ethicists from the start to the end, so not only at the moment of REC approval, that gene therapy is not only about informed consent and a favorable risk-benefit ratio, but also considerations of justice and fair access should play a much more prominent role in the ethical debate. And thank you for having me.
Thank you very much, Rieke. We have some time for questions. Are there any questions? I can't see all the hands up, so if you have a question, please just say it. Okay. If there are no questions, thank you very much, Rieke, for your very insightful presentation. And now we can pass to the second speaker on the ethical landscape. She's Phaedra Locquet. She has a background both in biomedical science, but also specialization in management and communication. And she's currently doing a PhD at KU Leuven with a focus on patient perspectives, specifically of patients with arrhythmogenic cardiopathy. Hope I said it right. And the value of gene therapy specifically for this condition. And the overall aim of our project is to develop a patient-centered roadmap for the implementation of this of gene therapy specifically for this condition. Thank you very much, Phaedra, for joining us, and the floor is yours.
Thank you, Alice, for the introduction. I indeed do research on patient perspectives towards gene therapies, and today I want to dive into that and share the perspectives of patients and the general public about what they think about gene therapy trials, but also why those perspectives matter. So to start off, this diagram gives us a useful overview. It shows that whether or not someone accepts gene therapies depends on several things: how much they know about it, what their personal or religious beliefs are, whether they are aware of the risks and the benefits, and to what extent they really understand those, but also where they get that information from. So very short, it's not just about the science. People's attitudes are shaped by a much broader picture. One of the key findings is that knowledge levels vary widely among patient populations. Many patients, or we see from the literature that many patients lack a basic understanding of how gene therapies work in general. For example, people are often not aware of what viral vectors are, which are the modified viruses that are used to deliver the genetic material into the cells. While these are typically engineered that they cannot replicate, patients still frequently worry about whether they might still cause or transmit an infection. Another area of confusion is the role of chemotherapy. In many gene therapy studies, particularly ex vivo gene therapy applications, chemotherapy is used beforehand to prepare the body, so-called conditioning. But patients do not always understand why that's really needed, and they might also underestimate the risks that come with this application, which can include increased toxicity, immune suppression, infection, organ damage, and compounded side effects from combining both treatments. As already highlighted by Rieke, there is also a significant gap in understanding timelines. Patients often struggle to grasp why there is such a prolonged interval between a scientific discovery and the start of a clinical trial, and then again before the regulatory approval. It's not just an abstract concern for patients whose condition is deteriorating. The slow pace of development can feel deeply personal. They worry that the regulatory timeline simply won't match their own, often narrow therapeutic window. And finally, there is also a tension in what patients say that they want. Many express a desire for strong, clear, and very detailed information, while others say they trust their healthcare providers and don't feel a need to really understand all the technical details themselves. Acceptance of gene therapy is variable, but generally increases after information provision. So this underscores the importance of patient education with appropriate information. We also see that there is more acceptance for patients with severe diseases and diseases associated with high mortality risks, as they are more willing to accept the risks accompanied by gene therapies. We also see that older people, male, and people with higher education show higher acceptance, while from the public perspective, we see that for older people there is less support for gene therapy. Interestingly, we also see that altruism is often a significant motivator, particularly among older individuals who may participate to benefit future patients. And in the case of genetic condition, it is often for the benefit of their offspring that might also be affected by the genetic disease. We also see some barriers to acceptance of gene therapies, for example, for non-therapeutic enhancement purposes. There we see a threat for genetic diversity in the long term, but we see when it comes to enhancement that there are more levels for support in terms of increasing lifespan, improving intelligence, and so forth. But as we know, this is not allowed. We also see some concerns among patients and the public when it comes to practical issues of affordability and access. They also report a need for trust in regulatory oversight, as patients want to know there are robust checks on science. In a number of studies, we also see that patients tended to overestimate the benefits of therapy, particularly around whether the therapies would be disease-limiting or disease-reversing. We also see that some studies report that patients were so eager to gain access to therapy that they expressed minimal consideration for the potential side effects and have unrealistic expectations around the timescales for the availability of the actual therapeutic option. Conversely, some patients are also worried about the considerable uncertainty around the benefit that may be derived given the potential risks of gene therapy. Particularly, parents of younger children express more concerns about the uncertain benefit risks balance, which appears to decrease from the moment that their children become older, the health of their children deteriorates, and the treatment options become more limited. In terms of understanding the risks, as already mentioned, there are some concerns about use of chemotherapy and gene therapy, given the association of chemotherapy with cancer treatment and the risks of developing cancer. Patients were worried that the side effects of chemotherapy would be taxing on their bodies and that they could be trading their current illness for cancer. In terms of information need and sources, we see that patients express a clear desire for information, particularly results from large long-term studies, data on risks and side effects, and personalized information such as eligibility criteria. We see that patient or people in general report to have gained understanding or some knowledge of gene therapy through a range of sources. They have heard from gene therapy through television, magazine, friends, and colleagues, but they do report that the most trusted source consistently remains their physician. The public also places high trust in scientists and medical researchers, which has direct implications for how we communicate about trials and for the responsibility placed on clinicians and researchers to provide accurate, accessible information. When it comes to patient involvement, we see that there is a growing recognition that patients should not simply be studied, but that they also should be involved more in research. On the slide, you can see a continuum of involvement moving from consultation, where patients are asked for their views on study design or consent procedures, through collaborations, where patients act as co-researchers in planning and data interpretation, to also co-production, where patients and researchers lead together with equal decision-making power. But why does this matter specifically for gene therapy? We see from evidence that meaningful patient involvement can improve trial design, recruitment, but it also can strengthen consent procedures, align the informed consent documents with information needs reported by patients. It can also make outcome measures more relevant. It also appears to build trust in science, and combining all these elements, it facilitates the uptake of new therapeutic approaches. So to conclude, we heard already from the previous talks that gene therapy trials are ethically distinct, because we have a therapy that comes with, in case of gene editing, particularly irreversibility. There is long-term uncertainty about the efficacy, but also about safety and side effects. And these therapies are often targeted for rare patient populations that might include vulnerable populations, which increases the demand for ethical scrutiny beyond standard drugs trials. We see also from the presentation from Rieke that the informed consent process is generally complex, because people might be prone to therapeutic misconception, but also the scientific complexity of gene therapy itself makes true informed consent in gene therapy trials particularly challenging. But also that we should involve patients from the beginning, because they have knowledge that is unique for their disease, and it can help us to understand what, from their perspectives, are acceptable risks and meaningful outcomes. Thank you. I'm also happy, before going further with the case, to take any questions.
Did you happen, I'm sorry, I just can't open my video, so I apologize. My name is Ilana. Did you notice any thoughts and fears of stigma, especially for long-term results? That's one thing. And the other thing, please, did you notice any reference to the literature of science fiction and etc., that many people now read? I mean, they've been reading this for a long time, but I wonder if there is any influence of that into the way people think about gene therapy.
So I think for the first question, when it comes to stigma, I think what we see, for example, for gene therapies that are targeted for hearing or people that are deaf, they report to feel stigmatized, because not for all patients being deaf is perceived to be a disability or a disease. So in that sense, people can experience stigmatization if they are being offered with a gene therapy that might cure them from this deafness. When it comes to your second question, science fiction. So I did interviews with patients that have a genetic heart condition, And there we did see indeed that they do report, okay, editing or having this therapy that really, tackles the genetic mutation sounds as science fiction from them. I really had one participant that expressed it in that sense, but it was not. It was more approached with being amazed by the fact that that was possible than being a barrier to receive or take on this kind of therapy in the future. So I think they see it as really something special that is even possible to correct the genetic mutation, but it's not per se mentioned. It was, or at least in my interview studies, not per se mentioned as a concern or a worry or anything.
Thank you so very much. Thank you.
Thank you. Any other questions?
Yes, Tina, go ahead.
Yes, hello. Thank you very much, Phaedra. That was very, very interesting, like all the previous speakers. Regarding your last slide about one of the last slides referring to these trials being ethically distinct from others, you also mentioned the issue of irreversibility. And could you elaborate a little bit more on that in relation to other kind of trials? Thank you.
Yes, I can. So I think, let me just, because, for example, particularly gene editing, as Pascal mentioned in his presentation, come with permanent modifications to the genome. And that is where the gene therapy effects may be irreversible because of these permanent genetic changes. From an efficacy, it can be considered positive because it's a once and done treatment in that sense. But from a safety level, there is no way to turn it off. So at the moment, there is no way to make the genetic to undo the genetic modification, while in more standard drug trials, they can stop taking the drug, and the effects will also diminish after a time. Also with a medical device, it's the same. While with gene therapies or gene editing in particular, there is no way to turn it off if something would go wrong.
Yes, if I may just ask a little bit more. Yes, I understand the technical point of irreversibility, but I was also thinking about whether the patients are really aware of that and how they perceive this issue when they participate in clinical trials. This is what I meant, actually, how they deal with this issue of irreversibility, whether it acts as a hurdle, as an obstacle, or whether they have this specifically in mind about that.
Well, so it was also a question that I had for the patients in my interview study. And for these patients with this genetic heart condition, the permanent nature of the therapy was not per se a reason to refrain from gene therapy, but they did consider the irreversibility when it comes to, okay, I would take on this treatment and let me rethink that a bit. So it was not a reason to refrain for patients, but when learning also about the fact that the efficacy might be uncertain on a long-term level, as well as the safety concerns. It was something that they took into account at that moment. However, in general, the irreversibility element specifically was not per se seen as really a reason to refrain. No.
Okay, thank you very much, Phaedra. Thank you.
Any other questions?
Yeah.
Okay, then I think, Alicia, we can go to the case.
Yes, I don't see any other raised hands, so I think we can move to the case.
Okay, okay. So I will present to you the case that we will be discussing in the breakout rooms. So here you can see an overview. So I will just out loud read the case. So over the past years, a team of researchers across the EU has been creating a gene editing-based gene therapy tool using a CRISPR-based gene editing tool. Sorry, there is a repetition in the sentence. To treat Leber congenital amaurosis. The team's approach is to create an ex vivo gene editing-based gene therapy to treat these patients, which will involve the following steps. So they will extract the retinal cells that express the disease-causing gene. These will be harvested from the patient's retina. Afterwards, they will be modified in the lab, where the gene editing intervention will be used to modify this disease-causing gene to restore the healthy function. Afterwards, patients will receive transplantation of these cells back into their retina. So the study will take approximately 24 months, and patients will be checked at regular intervals. It aims to recruit 25 patients aged between 18 and 40 years old, diagnosed with a specific mutation associated with the condition, and who already have significant visual impairment or blindness. The team conducted extensive preclinical studies in rats, which showed that this gene editing therapeutic provided significant improvement in eyesight. Team is hoping to determine the intervention safety, patient tolerability for treatment, and clinical efficacy. Finally, they have also stated that their intentions are only to create this product for therapeutic purposes and will not be creating intentional germline changes. So this will be the case that you will be discussing in the breakout rooms. I think Faye will now distribute all of us.
Yeah, before that, I would say, are there any questions on the case? Because some potential answer could be useful for everybody.
So since I don't know how long I can stay, things are happening around here, I just want to verify that extraction of retinal cells is a known procedure. It's a question mark statement. Is it a known procedure that can be done safely without causing further harm to the person? I got you. Okay. It's an kind of answer. Thank you.
Yeah, I can say something. I actually work in this disease and
okay, great.
with gene therapy. And so actually harvesting retinal cells is not something we do because it's not really possible to do like, kind of a biopsy and not having consequences. But I think for the sake of the case, let's not think that this is relevant. Let's think that it's just like the hematopoietic system where you can harvest cells, change them. But in practice, we really can't do this. Sorry to kind of...
Dr. Silva, thank you. I really appreciate this comment, but I do think that this was supposed to be raised because that's taking the first step based on something that does not have standard of care or regulation. So thank you for setting up the rule for the game.
Thank you. Vicky, I saw your hands up.
Yeah, one more question. How would you like us to treat this case? Would you like to treat it as an ethics committee that is reviewing such a protocol? What is being asked by the groups?
Thank you.
Yeah, the idea is that we will pretend to be the ethics committee that receives this protocol. And so what to look into this protocol, into this case study and see what are the main concerns that we would have, what could be the benefit, the risk, and ultimately, would you approve, reject, or let's say approve with amendments? So that is the idea. We'll also have questions to guide the discussion. Don't worry about that. But yeah, the idea is that to pretend to be an ethics committee. Other questions? Okay, then I think we can, Faye, if you can help us splitting us into the breaking rooms.
So I think before we start, so the idea is that we have a discussion, as Alice said, and consider to what extent we would approve or reject or approve with amendments this study. But before that, we need to have a note keeper that would like to also highlight after the breakout rooms in the plenary session what the main findings were. So is there anyone that wants to be the note keeper? Or yeah, or present the results later? Any volunteer?
Unfortunately, I'm going to be enjoying your company for the next 15 minutes and then I have to leave. So obviously, I'm not going to volunteer. Thank you so very much.
No problem. Anyone else? I see Alexandra, Robert, Constantina, and Sembra in. Sorry if I don't pronounce your names very well.
Me, I'm participating from Boston, so I woke up at 3:30 a.m., so I'm sorry, I may not be very effective.
Anyone else?
You know what, Federa, why don't you take the responsibility? Go ahead. Be the narrator and the scripter. Just go ahead. We'll support you.
Fedra, I can take the notes.
Oh, thank you.
Okay, that's great. So, from what you've heard so far, are there already some initial insights that you feel? What kind of aspects do you think that the proposal make it less or more ethically acceptable?
Hello. Mr. Robert Nurse, would you like to join the breakout rooms?
Yes, I thought I had already done that.
You're muted if you're talking to me.
Okay. Yes, I thought I had already pressed a join button.
You're still muted.
Robert, we can hear you.
Yeah?
Yeah, we can.
Okay, good.
So as an ethics committee member evaluating this research proposal, what do you think would this make this type of experiment more or less ethically acceptable? Anyone that wants to give some input on that?
Well, I think concerning the governance of the process, there should be more information about what would happen when there are unpredicted or unintended events, and would they act upon them without delay? So that should be also clarified in the very beginning.
So some safety measures
yes,
that they should implant along, or integrate in the clinical trial at several time points, and should we also do it then in the long term after the trial? Because I think, for example, There are, of course, some unknown long-term effects that we do not know from gene therapy. So I think also there's an opportunity to indeed have these measures integrated into the design.
And about the irreversibility issue just raised, what will happen if there is an unpredictable outcome? Would that mean the process will end or continue? If it ends without finalizing all the process, what would happen to the participant, the patient? Would that person lose anything which she already had in the very beginning?
Yeah.
So all those are risks to be considered beforehand.
Yeah, I agree, because of course we also have the thing when it becomes the right to withdraw from these kind of studies. But then I think, yeah, if we have gene therapy, which has irreversible implications, it's not that they can stop taking on this treatment. So I think indeed also there should be some strategies or support. I see Ilana.
Yes, I think structure is very important here. I understand that this is a research done all over Europe, and we are talking about 25 patients. I wonder if all the gene therapy are going to occur in one certain medical center that has all the facilities, or is it spread all over Europe? And I don't mind touring Europe, but patient needs to have access. And if it is going to be in several medical centers, they should all prove that they have the system, the capacity, and the duration for this protocol. Also, let me remind you, patients can stop participating in a clinical trial even if they went through a certain medical procedure, like having a stent or something, and they can drop out. So the question is the system made for people dropping out of the experiment or the process during those 24, and what are the protections? Another thing, I'm sorry, I didn't quite hear Sambra, but did you also refer to the issue of getting other medical treatments known for Leber syndrome? And are they going to antagonize the gene therapy? So are these people going to stop taking medications or treatments that are currently standard of care? And it should be stated out very clearly in the protocol. Yes, no, why exactly, etc. Also, in terms of long-term, sorry, once you're going through the experiment, long-term follow-up should be offered and given free of charge to those 24 people. So for me, it's like a yes, no signing on this protocol. I love this game. I love this game. It's great. Thank you, guys.
Thank you, Ilana. Anyone else that wants to comment on that? Okay, then I think maybe in terms of informed consent, do you think that there should be some specific elements that we really need to consider, or that the researchers definitely should have great attention for? Not specifically? Okay, I see Ilana shaking her head. Okay.
may I also say that I'm lacking like measures. When you're taking those cells out and you're doing the gene therapy, what is the percentage of corrected, sorry, I'm using my hands, but you can't see me, corrected cells that are going to be given back to the patients? Are we going to 100%, 70%, 50%, whatever? I need to know what the measures are and how the protocol is going to support this. But I love science fiction.
Anyone that wants to comment on that?
Hello. May I add that maybe patients should be aware of the previous preclinical trials on rats, so as they have in their hands some preliminary results, let's say. Maybe they will help them to consent or not to the trial, the therapy, actually, not the trial.
Yes, I agree. I think it's important that also from the preclinical studies that patients can maybe already have an idea of what the efficacy was there, but also what kind of side effects occurred there. For example, if we look back from the case from Jessie that was presented by Professor Rieke van der Graaf, for example, there the patient was not informed about 2 deaths that occurred in monkeys during the preclinical study. So I agree that it's very important also in this case that patients get this kind of information as well to really be able to make an informed decision in that sense as well. Do you think if we, of course, that's not the case, but if we would have this therapy for a pediatric population, where it would be able to treat the condition before the onset of the visual impairment or blindness, do you think that then there are unique considerations that the researchers should be aware of? Thank you, Ilana, for attending.
Yeah, I think it's absolutely necessary to adapt the way the information is given to this specific population that maybe can't read properly the informed consent, the information letters, and the information. So I guess they will have to be creative and maybe write in Braille or with videos and explanation given orally, I can imagine.
Yeah, I agree. I agree with that. I'm also thinking, because we earlier also discussed about the experience of stigma, which I'm not sure about this condition, but I can also imagine that there are also people that have or are blind that also don't really feel like they are really or experience it as a disability. So I think also there we also should consider this, that we also might be having patient or recruiting patients that might experience this stigma as well. I'm not sure. Do you how you feel about that? Is that something that you agree with, or?
And another point, maybe, since the pilot study was done on rats, I assume that they have rats, for example, may see better in dark than human beings. So what would what problems would that trigger when that is applied to human beings? So shall we start to see like rats better in dark, and how would that affect diversity? Would that pose some undeserved opportunities after treatment, which would threaten diversity, equality among human beings? So those may be other unpredicted outcomes, which lead to some negative societal outcomes.
Yeah. Okay, I think also that maybe also might affect the transferability or the way we can be able to interpret or translate the findings from the preclinical data to the clinical data, if indeed rats have different vision capacities. So yeah, interesting.
May I add another thing? What about patient involvement? Because the case study doesn't refer any scheme for consultation, collaboration, or production yet that you were previously referred to. Maybe we can add that note.
Yeah, I agree. I think, and particularly because, like Ophelia already mentioned, we have indeed a population that will not be able to read the informed consent procedure. So I think also there's definitely an opportunity to really tailor the informed consent procedures towards the needs of this patient population. And there, I think, as you say, patient involvement is a great opportunity. And also building on that, because I think what we often see now with gene therapy trials is that there are being a lot of visuals used to deliver information on what gene therapies are. A lot of videos are being made and so forth. But I think indeed that in these populations, this might be more challenging, and that indeed several measures will be required. Okay. So just in general, do you think that we should accept this treatment or this protocol, or if what kind of suggestions would you have that are still definitely needed besides those that we have already discussed? Do you think that if the gene therapy trial, the gene editing tool would be in vivo instead of ex vivo? So instead of that we harvest the retinal cells, that we inject the therapy directly into the eye, do you think that then the risks and the benefits might differ for patients? Would that maybe be a better approach?
In my opinion, I believe that this kind of therapy ex vivo is better than in vivo, because maybe the risks are more calculated than injecting the vector directly to the patient's body. I don't know.
No.
I don't know. What do you think? I think is more safe. The procedure is safer. I don't know. It's an allegation.
I agree with that. I think there are specific, and particularly in the case of gene editing, there are particular risks when it comes to really in vivo gene editing. But I mean, if we indeed then look at the question that was asked in the plenary by Ilana from the method of extraction of retinal cells, if that is indeed some procedure that might come with some, that is not standard of care and might also come with some adverse effects, it might indeed also be a reason to prefer in vivo. I don't know, but I can imagine that that also might be an element to consider. Okay.
And sorry, I don't know the science behind this technique, but is it possible also that if you do it ex vivo, then you can check the cells and if it worked, if the gene is well integrated to the DNA? I don't know.
I think for both, there are measures to indeed measure the expression of the new proteins that are being produced at that moment. So I think that's definitely possible in both.
Okay. Thank you.
Do you think that we should think about any issues related to inequity or access?
Yeah, I think we talked about checking if each center is well equipped and the capability of each center, but maybe also about the dissemination of this clinical trial, how to reach those patients. I guess this can lead to inequity if some countries or centers are better at identifying the patients, I can imagine.
Yeah, perhaps they are also, if we have a rare disease, I'm not sure if it's a rare disease, but maybe the condition is also more, has a high prevalence in a specific country that we should target, or yeah.
Indeed.
Okay. Anything else that anyone wants to add? Because I think we have discussed already several elements. Okay, Ophelie, do you have enough notes for the to present the findings of our discussion? Because I think we will be directly soon again to the plenary session. So
yeah, no, I think we have. I remember we had also a question maybe on pediatrics. Sorry, I don't know if you wanted to.
Yeah, I think I asked a question. So, but
yeah, sorry.
Yeah, excuse me. Okay. One more thing. We didn't mention chemotherapy. I don't know if it's involved in this particular gene editing therapy.
Well, it is. It's indeed not mentioned in the case. So, of course, it's a hypothetical case, but it's often is the case indeed with ex vivo gene editing therapy. So, I agree that chemotherapy might be relevant here. So, I'm not sure what you wanted to say about that.
Maybe we can address the researchers to include this in information, preclinical information to the patients. I don't know, what do you think? About the adverse effects and the possibility.
Yeah, I agree. I think definitely patients should be informed about that they indeed are not only receiving gene therapy, but that they will also be going through some chemotherapy beforehand as well. So I definitely agree that patients should be aware of all the effects that are associated with having this combination of therapies.
And also, a small detail, the case refers that patients will be checked at regular intervals. Maybe we can specify these intervals. For example, if the case is that there is one hospital that everyone is going there for the therapy, they should know in advance how regularly they should travel or what would be the cost.
Indeed. Because I can imagine that for them, if they have visual impairment, that they potentially might not be able to travel alone for a longer of time. So I think that also there that should be considered that we have indeed this specific patient population that needs to be able to get indeed to the sites that are having this gene therapy.
Yes, because we don't know if their impairment includes a caregiver.
Yeah,
who should travel along them.
I agree. Samra, anything that you want to add?
Yeah, when we focus on the last sentence, the team is hoping to determine the intervention safety, potential tolerability for treatment and clinical efficacy. It seems that the team is more interested in solving a research question rather than seeking to alleviate the suffering of the individuals. So is there another hope or target to improve vision of the patient, etc.? I don't see that. It's focusing on intervention safety, tolerability for treatment, and clinical efficacy. Does it involve well-being of the patient, improved enhancement of vision? So these are not very clear in the purpose statement, I think. It should be more clearly defined.
I agree. I think we should have indeed a bit of a sense of an idea of what the outcomes are specifically that they would like to measure. And indeed, if those outcomes are important also from a patient perspective. So I think their patient involvement can also play a role again. I think of course, in the earlier phases of clinical trials, the research question is indeed mostly aimed at safety and tolerability. But I think, I agree with what you say about that we have to have a better idea of what the specific outcomes are that they are going to measure when it comes to efficacy.
Because this doesn't only somehow ensure that, the voice of the patient is heard, but they can even make a protocol better from a scientific point of view, because they actually know what it takes to live with the disease, and they can also provide feedback about whether in this period of 20 months of the clinical trial, it's possible to follow up all the visits, whatever is included in the protocol. So if they participate in this procedure, they can make the whole trial better and have their voice heard as well.
Yeah, thank you. Do you think that for the same logic could be also useful to have patients' involvement also in the development of the information material?
That would be very useful. Also because, sometimes we as researchers, we have a certain idea of what would be important to the patients, but we are not patients. So we really we really can't put ourselves in in other person's shoes. So that would be very, very interesting.
Yeah, thank you.
If I may add one more point that is not related. I've been thinking about these 25 patients, like the sample size. Could it be that for a first in human trial, this is a very big number of patients? And the other thing is that whether there will be like a dose escalation between, let's say, the first patient and the second, and then the third, and so on. Because, and what time intervals will exist between the different patients? Because we can take quite, regarding safety, it might be important to allow for some bigger periods between the different patients participating.
I can add a little bit to that. Usually, if it's like first in human, I would think 25 is too much for a single stage. If, like Vicky was mentioning, if you kind of split them, I would probably say, 9, 9, 9, something like that, or 8, 8, 8. And you have a time lag between the first few patients to see if, if there's no issues before moving on to the second group. And maybe just the dose escalation in the last group, something like that. So I think that, yeah, I agree with you. 25, like, for a first approach, given that this is actually a rare disease, it's probably a bit too much. Yeah.
Yeah, I agree. Let's do another imagination effort. Again, let's move a step forward. So considering that this is, from my understanding, something that doesn't happen actually later in life, but is a disease that occurs at birth or very early after birth. So what if the decision was that, well, maybe this could be more, let's think it's already, you did already a phase 1. So we moved a little bit further and we say, well, this could actually be really, really beneficial for children though. Then what could be the issues there? And would you approve it for children?
I don't want to be the single person talking, but for me, yes, I would probably move to younger patients, because we know that the younger these therapies are administered, the greater the benefit, and even the preservation of the vision and recovery of some vision. I think that the main issues here are the expectations, both of parents and of kids, which would obviously be very, 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 very high. And I think that in this case would be even more relevant, all the measures that we already discussed for not creating too many expectations, especially if you already have some, encouraging results from phase 1, like we are working here. So that would be even more challenging to kind of manage the expectations at this stage. And I think that probably would be even more important to involve patients or pediatric patients and their parents in the design of the study, and and giving feedback on the study, because so that people wouldn't get too excited and having higher expectations.
Yeah. Any others? But also, if I understand correctly, the idea is that you would approve it for children if it's, let's say, a phase 2 or 3, but not if it is a phase 1.
Not at the beginning, at the real start. No, I would probably start with adults, because in a way, usually they reach, or the people that suffer from this mutation, when they reach an adult life, they're pretty much fully blind. They have almost no residual vision. So in a way, that would be the safety trial, and especially because here we're thinking about something that in theory cannot be reversed. So, when you are, an adult, you can consent and you are changing something that's your own body. But with children, that this would be the parent's decision. And I think that this would also create a lot of, stress or anxiety in the parents because they have to consent on something that in principle would not be changeable, right? Or would not be reversible. So I think that this would probably need the support of more data so that the decision could be, at least a little bit more informed.
Yeah, I agree. And also, if I may add, something else that I would consider is the fact that exactly because this is a permanent change, if it was a phase 1, then I can imagine that you would have a lot of follow-up throughout your life, or at least for a huge chunk of your life, to be really sure of what will be the changes long term, etc., etc. And one thing is an adult, this like agreeing to that and being sure. One thing is an adult like consenting for a child, and then it's very restrictive, might be perceived as very restrictive for like the child life if then they have to go throughout all this follow-up without actually having consented to do so. Vicky, did you want to add something?
Yeah, but sorry, I'm sharing the office with somebody else. So
no problem.
Apologies. I can add my comment later.
It's okay. Yeah, fair enough. And I do have another question. What would happen in the case in which, for example, the parents want the trial, but the child doesn't? Or the other way around?
I mean, that's really tricky. I would say that it would probably need to involve a lot of talking and a lot of discussions before actually any decision is made. Because, I mean, if the parents want and the kid doesn't want, I mean, supposedly we cannot force the kid, right? But if we think that this is for their benefit in the long term, I mean, I think that this would probably involve a lot of, I don't know, child psychiatrists or psychologists or something like that, and probably have like a team that can discuss this. And because we as researchers, we see, the possibilities and the positive outcomes, but I think that this needs to be kind of translated by someone who's more accustomed to deal with kids and parents. So maybe adding people to the team that can moderate this discussion and see if they can reach an agreement, either participating or not.
Well, if I may, this is an age-dependent issue because we cannot generalize. There are different age ranges, and then that would differentiate the approach. But we do need the child's assent for participation in the trial, so we can't give a generic reply to this. It depends also on the age and the narrative of each child, but we certainly cannot force a child to participate into such a trial.
Yeah, I would say maybe there would be a difference if the trial was life-saving, potentially life-saving, let's say, but in this case, it's a bit different. And if should it work, in principle, could do it later on in life. So, okay, I have one semi-last question before going back to the plenary. So at the moment, we really just talked in terms of like the trial, what are the issues on the trials and so on. But again, let's take it a step further and imagine that the trial goes well. And we have a therapy now. What do you think could be some some issues more related in terms of access and inequity. Marco, this is your topic, so feel free to jump in at any point.
I mean, this would be very expensive, for sure. And have it being a rare disease, I mean, the population that you would have to intervene would be smaller. If we think that, you know, blindness is a debilitating disease that greatly reduces quality of life, I mean, that would probably be very important that the, you know, national health systems would step in. And I always think that, you know, if they would probably do like a joint thing, maybe that would be easier than to just each individual country try to get, you know, these new novel therapies. But I mean, it's an expensive, and it's going to be expensive, and that would probably be a very big barrier to the implementation or to making this therapy available. I was thinking on the lectures, and you know, probably, you know, they will probably be able to get this in you know, in one of those exemptions or something like that. But it's still the cost would be probably very high, and that will create a lot of you know, inequality in between countries. And I don't have a real solution for that, unfortunately.
But also, I would say within country, should it not be paid through the NHS? Then also inside the same country.
Yeah, I totally agree with that. So the national health systems would probably have to step in for this. And I mean, I speak about my own reality here in Portugal. Usually they do, and sometimes when they really don't do it as fast, there's also some, you know, media pressure and family pressure through the media, and that usually kind of helps a little bit. So indeed, I think that for at least to try to achieve some sort of you know, equal access or equal opportunity of access, it would definitely have to be through a national health system.
Anyone else? Vicky?
Yeah, I would like to add that there is an equal access even between countries at the European level even. So, for example, countries with high R&D, they are in a better position negotiating prices, etc., directly with the pharmaceutical industry, compared to countries which do not hold so many resources or are not that active in R&D. And we do have examples, you know, practical examples from the past. So I think that perhaps what was mentioned earlier, I think it was Cynthia that mentioned it, we should see at the European level whether we do have a better negotiating capacity as EU compared to individual governments and nations. So this is a huge issue. And the pharmaceutical companies developing gene therapies know that it's a high cost for most of the national health systems. So they're trying to also find ways, and maybe this could be one way, I mean, just as a suggestion.
Mm-hmm. Yeah, indeed. Also, Vicky, if you want to add the comments from before, please feel free.
You mentioned earlier that if this clinical trial was also recruiting children, by law, according to the regulation of advanced medicinal products, children have to be indeed followed up for longer periods compared to adults. So this is something that they should be, the children and their parents should be aware of this, that when they participate in gene therapy clinical trials, they should be aware that we talk about long-term follow-up here.
Mm-mm. Yeah, indeed. Okay, anyone else?
I just remember something.
Yeah, go ahead, please.
Actually, I think from the previous training, which was that we could use virtual reality for children to understand a little bit what would happen. So maybe design something like that. So I remember that
we
from
were in the same breakout room. Yes. Yeah, that could work, but I don't know how easily it would work with a child who's blind.
Yeah, well, I was thinking maybe something tactile or something,
Yeah,
that they could
it had to be
feel.
like
I don't know. I'm not very good at,
but I
artificial
think it is
intelligence.
feasible. Like it could be a different form of virtual reality, in which is
Yeah.
more like audio and audio
Yeah.
sensory, like more than visual
Yeah.
based.
Yeah.
But I don't think it's impossible. But I think, yeah, indeed, for children could be useful to give them a clear idea of what's actually going to happen to them.
May I also add one other aspect that is
Yes, of
coming
course.
again and again in my mind? I'm thinking we're talking about very serious diseases which are treated with gene therapy that are quite complex clinical trials, etc. I'm thinking if there is an overlap between different ethics committees that will be reviewing such a protocol for each country. So I'm thinking that perhaps in Greece, it would be just the National Research Ethics Committee at the national level. But I'm wondering if there are other kinds of overlaps in other countries that make things even more complicated. So, and whether this challenges the actual start of the clinical trial in any way.
Yeah, I think that is a massive issue when it comes, especially to this kind of trials, because a lot of time they're rare diseases, right? So you a lot of time you don't have all the population you need in one country. It's a cross-country trial, which for some reason it's a great thing. But then from an administrative point of view, indeed, it takes a very, very long time just to get the approval because it's a lot of different bodies that you need to go through, and the rules might change slightly from country to country. So something might be okay for one country, but then has to be adapted for a different country. So yeah, in terms of administration, it's long. I mean, it's good on one hand that we are so thorough on the ethics, so I'm not going to complain about that. But yeah, indeed, it's something that a lot of time, I think, in this trial, they take into account that I think 1 year minimum just goes, if it's a multi-country trial, just goes in administration and be sure that everything is in order, that all your documents are compliant and so on. And of course, that makes the trial even more expensive.
And by definition, if we talk about rare diseases, it has to be multinational or multi-center at least. So that creates more issues. Yeah.
Yes, indeed. And it's more issues like from let's say, from a bureaucratic point of view, as we were saying, but also for the patients. Well, I say patients because in these situations, participants are patients most of the time as well. So they have kind of a dual role, which makes things sometimes more complex. But also for them, because a lot of time it's not a treatment that you will have available in every hospital. So you will have more likely than not to travel to get to the center that offers the treatment and so on. And that also can add costs either to the patient or to the trial, depending on who is covering that cost of like the travel and if you have to stay there. And of course, in case you have children, it's not just a child, because of course you're not sending a child alone on the other part of the country. So more than likely it's like at least the child and an adult that will have to travel and have a form of housing. So that's also, it adds cost, but also it adds burdens for the participants as well, because it's days off that you need to take from school or from work. So it comes to a lot of, let's say, more practical challenges, but that's for a trial like this can become real burden for the participants as well.
If
Yes,
I could
Vicky.
add something, I'm reading this last sentence of the case that the team says that they will only be creating this for therapeutic purposes and there will be no intentional germline changes. So for safety reasons, we know that these gene editing techniques might also cause the so-called off-target effect. So there might be some changes in other parts of the genome, etc. So as an ethics committee, I would really like to also see the follow-up of the protocol for following up these patients, and that should also include analysis or in order to identify any potential off-target effects in any other parts of the genome. So the follow-up protocol is equally important as the recruitment at the beginning of the clinical trial, I think.
Yeah. Yeah, indeed, I agree.
And I would also like to ask, sort of point out, but also ask whether this kind of participants require some psychological support during the trial, either because their hopes are not do not come true, or because if there is some success, there is an important change in their lives, or how they deal with that in their families, or if they're old people and they've been sort of used to a certain way of life. Maybe Gabriela would know something about that. I'm just thinking loud.
It's very important. I see there hands raised from Faye. I think Faye,
Yes.
it's time to go back to the plenary, isn't
Yes,
it?
I'd just like to inform you that the other room has went back to the main room. So if you're okay, I can close the
Yeah,
rooms.
we can conclude. Thank you very much. It was a very nice discussion. Okay, are we all here, I think? Thank you, everyone. Now it is time to kind of wrap up, but before, it would be nice to have a little bit of a report of the key messages or the key points that have been discussed in the different breakout rooms. We can start with breakout room 1, but I honestly don't remember which one is 1 and which one is 2.
We were 2.
Ophelia, you were 1?
Yeah, I think with Fedra we were 1.
Okay.
So we discussed about many things. First of all, the safety measures that should be integrated throughout the clinical trial at different points, including long-term follow-up. We think the participants should be offered long-term follow-up, and also that they clearly understand the efficacy, the possible side effects, and all the procedures involved, including the fact that they may receive chemotherapy before the gene therapy itself. Also that previous trials on rats should be mentioned as well. Because this population is specific, is visually impaired, informed consent has to be adapted. So information should not rely only on written information letters, but also include formats such as braille or audio, video descriptions. They should be informed in advance about maybe the organization and planning of the information, of the intervention, maybe they need to travel with a companion, and transport, accommodation can be maybe a bit more complicated. So maybe, also reimbursement of cost should be mentioned maybe. They should also be able to withdraw from the trial at any moment. So the details of what this means in practice should be clarified. And access and equity also. So will these gene therapies only happen in a few specialized centers or be spread across Europe? And maybe the capabilities of each center may be demonstrated. And also about access and equity, maybe the dissemination and recruitment strategies also matter, especially for a rare disease. We're not sure if it's rare, this disease, but like how will patients be identified and reached, and maybe some countries or population be more targeted than others. And finally, patient involvement could also help define more relevant and meaningful outcomes for the trial.
Thank you very much, Ophelie. For group 2, Tina, thank you for volunteering.
Yes, thank you. Well, we discussed the following points. So we started from the need for the study to be morally and ethically acceptable for a proper recruitment of patients. There was some discussion on whether there should be younger patients given the scientific aspects of this disease. And fewer patients because the number seemed rather large for starting such a study. In case younger patients participate, then informed consent should include the fact that they should be followed up according to the regulation for longer periods, and both parents and children and younger patients should be aware of that. Regarding safety issues, there was a discussion about the sufficiency of using rat models for this study. One participant, Vicky, mentioned that maybe they should have studies on non-human primates. We were concerned about the level of safety with the description of the study as it stands regarding the preclinical data. The issue was the preclinical data existing. Then we came to the discussion about the source of the material and harvesting the retina cells and whether this is a safe procedure or not. We discussed a scenario of retrieving the material already existing or from cadaveric tissue, which may not be scientifically possible regarding to what Gabriella said. We talked about the possible barriers, and these include therapeutic misconception and the strategies for dealing with all these issues. We came up with a conclusion that these people are not particularly vulnerable, but they do require, as was previously said in group 1, specific forms of consent and provision of information. And I think that was all, so my colleagues can fill in the gaps I left.
No, I think that was a pretty comprehensive summary. Thank you.
And we also talked about the inclusion criteria, that they have to be very carefully described and designed regarding therapies that these people have already received, or whether this study is proposed to them as a last resort, and this has to do with their psychological attitude towards the study and towards the provision of informed consent.
Yeah, indeed. Thank you. And there was no group 3, correct?
Exactly, only 2.
Only 2, perfect. Then we are back on time. This is amazing. We can wrap up the session. This was the end of our session. Thank you so much for being here, and thank you so much for your engagement and your participation, especially in the breakout room. It's always nice to have a very nice discussion. So thank you very much, and hopefully we will see each other in the next training sessions that will happen in autumn. We don't have specific dates yet. You will receive more information later on. Thank you very much again.